where chemical characterization of antibody was done, right? Okay, so did you understand what was discussed in this slide? Or it was not clear?
Sir, could you give a brief overview once again?
Yeah, all right. So what they did was, so basically they injected this model antigen, chicken ovalbumin, in rabbits, and after a few weeks time, when the injected antigen generated some response in form of basically activating your B cells, and these B cells made antibodies. So that serum sample was collected from these animals. Okay. Now you have the serum sample which is enriched in antibodies specific to chicken ovalbumin, right? Now the question was, in which fraction these antibodies are present when you subject the, because serum actually is a lot of proteins, right? Serum actually is nothing but everything is proteinaceous in serum, right? So if you subject this sample to resolution under the influence of electric field, okay, so you get these different protein fractions separated depending on their charge, right? So what they did was, so basically they had the serum, right, as is shown with this blue line. Basically you loaded somewhere, for example on the slide, the sample, and then you subject it to electric field. Now depending on the charge on different fraction in serum, so these fractions were resolved in one peak, second peak, third peak, fourth peak. This first peak was very close to this positive electrode, right? And the other fractions actually were alpha, beta, and gamma globulin. All right. So now the question they were trying to answer was, in which fraction do you have something which is induced in animals as a result of this immunization process? So what is increased? What fraction is increased? And in order to do so, what they did was, so they basically incubated. In one case, you have the serum, the complete serum. In another, you have serum added with the antigen. And once you have this complex, immune complex formed, so you centrifuge it, because these complexes are going to be heavier. So they will settle in the bottom. After centrifugation, you collect the supernatant and load that supernatant, right? And if you try to overlay these peaks that you obtain by quantifying the content of protein in different fractions, so you can clearly see that following adsorption, this process is called adsorption, where you have added antigen with the antibodies. Antibodies were present actually in the serum, and then you pull them out, you remove it, you deprived your serum of ovalbumin specific antibodies. So it is not going to remove every single antibody. So that is why some fraction is still there, right? But the antigen specific antibodies were removed, and those antibodies actually were migrating in the gamma region. And by definition, this this protein was globular in nature. There are different forms of this protein. Okay, globular in nature, migrated in gamma region under the influence of electric field, and because this fraction showed reactivity against the antigen, so this is called immunoglobulin. Now, because this is close to the negative charge, so that means more or less it had positively charged ion. Matlab, the net content of charge on these antibodies is positive. Okay, so this is what they showed here. Using a very simple technique that we call as electrophoresis, performed on surface of a slide. You have a slide, you put agarose, you load the sample, you put these electrodes, and this will resolve depending on whether it is positively charged or negatively charged, whether it's more positive, more negative. So you get the resolution. Now, they found out that these antibodies were present in the fraction, which actually is very close to the negative electrode. Right. So based on this, they said that the immunoglobulin fraction actually is of this particular charge. Okay.
Excuse
Now,
me, sir.
yes.
Sir, in this absorbance assay, we know that the different types of antibodies, the 5 types that are present, so
Yeah.
they are actually the gamma type of globulins. So, sir, were they able to further resolve this absorbance assay, or were
Well, no.
these
At this time, no. Okay, so this was actually the very first experiment to determine what is the nature, what is the chemical nature of these antibodies, what charge do they express on their surface. Okay, so though the other things will come later. So that is what we are going to discuss step by step.
right, sir?
Sir,
yeah.
Sir, in the gamma fraction, we have only the antibodies, or
no?
are they
So there were some other things as well. So we will talk about in next slide. Okay, so basically, what are what is the main composition of alpha, beta, and gamma globulins?
Answer.
Okay, so the technique that was used is called immunoelectrophoresis, right? And we discussed about this alpha globulin, beta globulin, gamma globulin. Okay. Now, alpha globulin were the protein which were further distinguished depending on their migration and their nature and their size into alpha 1 and alpha 2 globulins. Alpha 1 actually is this antitrypsin. This is an enzyme which actually is predominantly present in your lung and liver tissues. Okay. Now, I don't know whether actually you are following the research done on coronavirus right from the very early days, starting from 2019-2020. So one group from IISc, they showed that somehow the level of this antitrypsin present in lungs can determine the severity of the disease. And why is it? Because trypsin is an enzyme. Actually, it can also promote the infectivity by this virus. And how does it do so? This is a little more complicated, and I don't actually expect you to understand everything. But somehow, so on the surface of this virus, you have this spike protein, right? At least this much is known. Everybody knows this thing these days, right? So you have this primary structure of the spike protein on surface of this virus, right? And in this region, in the neck region, so basically you have a site. And this site actually was in news for quite some time, and it still is debated whether this is a manufactured virus or this is a natural virus, whether this is coming from a lab or it's coming from the jumping of species from bats to humans, right? And one of the tipping point was this site. Okay. Initially, it was thought that this site was not present in any of the coronaviruses, and it was acquired by this newly formed coronavirus, but now we kind of know that this issue is more or less settled because some of the bat coronaviruses also have this site. So that means actually this theory, which suggested that this migrated from bat to humans, is more relevant. Okay. Now, why I'm bringing about this site? This is called furin cleavage site, also present in many more viruses. This is not unique to this coronavirus. Okay. Now, this site is cleaved by many of these proteases, which are actually present in lungs. And one of them you might have heard about is TMPRSS2, right? Which is present on surface of these cells sitting in your lungs. Now, there are other enzymes like trypsin. So that can also actually cleave this site and it promotes infectivity, right? Now, if you somehow sequester the level of this trypsin by antitrypsin, so the infectivity will be reduced. And it was kind of suggested that somehow the individuals who do not develop severe disease, so they also have a quite significant amount of this antitrypsin in their lung tissues. Okay, this is just to put things into context. Okay, these are not something that we actually are discussing out of nowhere. So these proteins are actually immunologically relevant, and they do have a role in promoting or inhibiting virus infectivity or the pathogenesis of other diseases, right? Alpha-1, one of the examples of alpha-1 globulin is antitrypsin. Alpha-2 globulin, one of the example is haptoglobulin. It's a protein that binds to hemoglobin to prevent its excretion. Okay, hemoglobinuria is a condition. Okay, where basically you get blood in urine. Okay, and that somehow is controlled by this haptoglobulin. Okay, these are actually clinically relevant proteins, alpha-1 and alpha-2. So you find a lot of these alpha globulins induced during inflammation, tissue damage, autoimmune disorders, and certain cancers. Okay. Now, beta globulins are these proteins which bind, which are actually these low density substances involved in transport of fat, iron, and so on and so forth. Okay. These are lipoproteins, transferrin, plasminogen, complement components. So these all are actually part of beta globulins. Gamma globulin are antibody we already talked about, right? They are majorly involved in immune system. Okay, basically during infections, allergic reactions, organ transplantations. In many of these things, actually you have gamma globulins. In other words, immunoglobulins, if they bind to the antigen, right? So those are clinically relevant molecules in alpha, beta, and gamma globulin. And in this table, basically here, so the concentration, the relative concentration of these proteins is shown, which is actually present in serum, right? Total proteins you find normally, which is like about 8 grams per deciliter of your blood. Albumin is quite a lot of it actually is quite albumin. And then you have alpha globulin, alpha-1, alpha-2 globulins, which are actually is 0.15 to 0.25, 0.43, 0.75. Beta globulin are 0.5 to 1, and gamma globulin actually is in this particular concentration, around 1 gram per deciliter, right? So just to give you an idea, so what is the relative representation? And based on this, you can actually also find out this thing here. So you are looking at the absorbance value of the resolved protein in this area, this area, this area, and this area. And the size of the peak would tell you the relative concentrations, right? Albumin actually is much more. Gamma globulin is second, and then you have the alpha and beta. which is actually also shown more or less somewhere here. A large fraction of your serum actually is albumin, and the rest of it actually is constituted by beta and alpha and gamma globulins. How these things are resolved? By this technique that we call as immunoelectrophoresis. Maybe I think in one of the lectures I'll talk a little bit more about this immunoelectrophoresis. But for the time being, just remember, using this very simple technique, you can actually find out a lot of information from which is actually clinically relevant. All right. Now, if we move on to antibodies, so these are basically called gamma globulins, right? In other words, the other name of these antibodies is immunoglobulin, because they show this immune reactivity, right? They come in 5 different flavors. Rather, these are called isotypes of antibodies: IgG, IgM, IgA, IgE, IgT. Now, what decides whether this particular antibody would be called as IgG, IgM, A, or E, or D? The heavy chain constant region. Okay, so in this you have a gamma chain, in this you have a mu chain, in this you have alpha chain, in this you have epsilon chain, which is the heavy chain of the antibody, right? And here you have the delta. All right, so these are 5 different isotypes of antibodies. Now we are going to talk the general structure of antibody molecule. So antibody molecules, they contain 4 polypeptide chains. Okay. Essentially, this is a dimer of heterodimers. Just understand it. It's a dimer. So that means you have a monomeric unit and combined 2 times. Monomeric unit is basically 1 light chain, 1 heavy chain. Right? Now, these are not identical. And that is why this is called hetero. This is not homodimer. This is heterodimer. You have light chain, which has these 2 domains, and heavy chain, 4 domains, or sometime you also get 5. Okay, so that really depends on the class of antibody you're talking about, or the isotype of antibody you're talking about. Okay. A dimer of heterodimer. Is that clear? This is a heterodimer, and if this is combined through heavy chain with the other heterodimer, so this constitutes a dimer. Okay, now what is the composition of light chain? So you have something that we call as V. V stands for variable, and C stands for constant. L stands for light. H stands for heavy, right? And if this is H1, H2, H3, so these are different domains. Okay. Now, broadly, you classify antibodies into 2 regions. One is basically the FAB, fragment antigen binding, fragment antigen binding. And the other thing that you actually get is FC. C stands for, this is not the same C. Okay, so this is a constant, and FC means fragment crystallizable. Okay, so this fragment, the constant region actually can be crystallized very easily. So if you want to determine the crystal structure, the hardest part to generate a crystal structure is on the variable region. Constant region can be crystallized in a much better way. Okay, FAB and FC portion. FC portion is predominantly involved in causing the biological activities. We'll talk about what those biological activities are. And FAB portion is the region of antibody which actually bind to the antigen. So essentially, antibody is a Y-shaped structure, and at the tip of these basically a Y-shaped structure, you have these two paratopes, one of this, and then you have the stalk region. This is basically the constant FC portion. This portion is FAB. Now in FAB as well, basically you have this variable portion and the constant portion, right? So this is what is shown here. So you have, let's talk about the light chain first. So you have these two domains. These are two domains in the light chain, VL and CL. One is variable domain of light chain, constant domain of light chain. And these, why we call them as domain? Because they are actually separated as a bulged part because of the presence of these disulfide bonds, or whatever is shown here by this yellow line, right? And this portion actually is also called as immunoglobulin fold. This portion is also called as immunoglobulin fold everywhere. A stretch of amino acid, which is usually about 100 amino acids, and then you have this formation of this disulfide bond, and whatever this fold that you get, this is called immunoglobulin fold, this portion, right? Okay, so the same is the case with the other light chain. Heavy chain, so I'll get back to this variable region again. Okay, heavy chain, so again you have one variable domain, variable region of the heavy chain, and then you have the multiple constant regions like CH1, CH2, CH3, and these are basically the intra-chain disulfide bond. Now heavy and light chain are also connected by this interchain disulfide bonds, and heavy and heavy chain are also connected by interchain disulfide bond. All right. Now let's talk a little bit more about this variable region. So why we call it as variable? Because you can have the same constant region in one antibody molecule produced by a clone, the expanded clones of a lymphocyte, a B lymphocyte. Okay, so the constant region remains same, but the major changes that you see are in the variable region. Okay, so what that means is it's highly likely that if you have two B cells isolated from blood, so these two B cells, maybe they are encoding for the same constant region, but the variable region is going to be different, right? And that difference in variable region means that this is going to bind to a different antigen. This difference, it actually can be of one amino acid. Okay, even if you change one amino acid in the variable region, so it actually changes the specificity of the antibody. And this is the process actually by which you can generate billions and billions of diversities. Diversities means specificities of antibodies which are going to bind to one antigen, second antigen, 1 millionth antigen, 1 billionth antigen. So all these things are brought about by the variable region of heavy and light chains combined, right? Now, even within this variable domain, so you have these purplish structures. Okay, in light chain, so you have three. Also, actually, in heavy chain, you have largely three of those. So somehow it's shown four. This fourth is not well defined. Okay, in many clones, if you analyze, so you don't really find the fourth variable region. So largely, you call them as three in light chain and three in heavy chain. Now, these regions, we call them as hypervariable regions. Hypervariable regions. So we will again talk about why we call them as hypervariable, because if, for example, you sequence 100 or maybe whatever millions of antibodies isolated from an individual, okay, so the major changes in amino acid composition will be in these regions, in three defined regions in light chain and 3 defined regions in heavy chain. So these are basically, they will vary hugely from one antibody molecule to the next antibody molecule to the other, to the fourth, to whatever number you go to, right? So that is why these are called hypervariable regions. 3 of them are present in heavy and 3 of them are present in light. Ignore this fourth one because this is not really well defined. Some aberrant observations might have suggested that there is another region which is more variable, but 3 are well established. Okay. Now, there are other things that you need to remember as far as the structure of an antibody molecule is concerned. Okay. So the portion where you have this FAB and FC connected, so this is called hinge region. Hinge. You know hinge. So you must have seen these doors, right? So basically, let's consider this. This is actually a choke hut, right? I don't know what you call them. But this door can move around with a fixed whatever pillar. Okay. And because this is able to move around, and this move is because you have used this hinge. Okay. So because of the movement of this portion of your antibody, so these actually can move around, but this constant remains same, right? So it's not like they will completely open up and completely close. So there is a certain allowable limit in which they can actually move around. And this is to ensure that these can bind to different antigenic determinants or different epitopes. So sometimes these epitopes might be present very close, so you have to basically do it like this. And sometimes you have to actually spread them out, sometimes even more, right? So that flexibility is provided by this hinge region in the antibody molecule. And now you can see that the composition of amino acid in the hinge region is going to be different. So you might have quite a few glycine residues here. Glycine actually is quite flexible, right? Now, the other thing that I said or told or actually mentioned very briefly was that antibodies are also very critical in activating one arm of your innate immune defenses. And that innate immune defense branch is basically the complement activation process. There are 3 different pathways, but the classical pathway of complement activation is brought or is activated or initiated by the antibody molecule. And why is it? Because antibody molecule in this region, in CH2 domain, has a site which can bind to the component of complement system, particularly C1q. So this, we'll talk about what are different components of this complement system are. But these complements actually components can bind to this domain. And following this binding, so you induce the activation cascade of the complement system, right? And this portion also has a lot of these glycosylated sites. Okay. For example, asparagine residues are quite a lot, which are predominantly present in the CS2 domain. And these can be N-linked glycosylation and O-linked glycosylation, right? So a lot of carbohydrates are present in this portion of the antibody. Carbohydrates, which is actually shown with this little structure here. right? What other things I need to tell you about this? Complement binding I have shown. CS2 domain actually is pretty important. It has complement binding site. It is also having this glycosylated region. And above this actually is this hinge region, actually directly above this disulfide bond. Okay, so this portion actually has the sequence of those amino acids which are more flexible, right? Now, so right above this disulfide bond, so there are also these residues which can be cleaved by this enzyme. There is a relevance of having these sites. And this is called papain cleavage site. Okay, papain is basically a serine protease again, right? And this enzyme was used. Why we call it as papain? Because this is actually you find a lot of it is in papaya fruit. Okay, you can get this enzyme, and if you basically incubate this enzyme in appropriate buffering condition with the antibody molecule, so this is going to cut here. And if this is going to cut here, so you can actually separate FAB region with from the FC region, right? And this is how it was determined that the antibody binding site actually is present in the FAB region and not in the FC region. Okay, now on the other side, basically, this is written all these different isotypes or different classes of antibodies. Class and isotype basically is one and the same thing. Okay. For example, in IgM, this region actually, this constant region, heavy chain is mu. In G, this is gamma. In IgA, this is alpha. In IgD, this is delta. And IgE, this is epsilon. Okay, so this portion is changing. All other things might remain same. IgM, IgG, IgA, IgD, IgE can have the same antigen specificity. They can actually bind to the same antigen. This is actually quite logically true that all these antibodies can have the same antigen specificity. Okay, but because they will be different in the constant region of the heavy chain, so these will be called as IgM, IgG, Ig, whatever, ADE. So any clarification from this slide? Because actually this is very important slide if you want to know the structure of antibody, and almost for most of it, we are going to talk about the antibody in a major way.
Excuse me, sir.
Yeah.
Sir, generally when we see a structure of an antibody, we generally see that the light chains are positioned below the heavy chains, whereas here they are upwards.
Well, okay. Well, you don't worry about that. Okay. So it's sometimes people can show them basically that you have the heavy chain and then light chain here on sometime they can actually be displayed on the other side. Okay. So this actually really depend on how you are looking at the structure of the antibody molecule. Okay.
So sir, I was just I just wanted to know that in both these structures, whether there will be any steric hindrance if we consider both types of structures.
Well, not in terms of well, obviously, and that is why basically this region. So even let's say you have one chain and then you have the two chain. So this chain can keep on actually flipping around, right? And this is basically nothing but the amino acids, and these amino acids have a certain movements, right? So they can actually adjust to wherever they have to bind.
Right, sir. Yeah. Yes, sir.
So it's not like don't think that this is a basically stone-walled structure. Okay, it's a very flexible structure. It can keep on moving around, right? Yeah, it actually depends on whether you have a long hinge region or a short hinge region. That will define the movement, whether you have a less or more. All right. So
Sir,
yes.
sir, here you told that the papain cleavage site helped in actually separating the FAB and FC and thereby actually helping to know that FAB is actually binding to antigen. So my question is that during their experiment, I mean, whenever it was discovered, how did they do that? Because I think without the FC region, FAB can't alone bind to antigen, right, sir?
Well, no. For antigen binding, you don't require FC region. FC region is for other things. For example, if it has to bind to FC receptor, if it has to move from one site of, for example, transport of these antibodies, this is called transcytosis. So basically, for example, in intestine, from one portion, here these are produced, but they have to get into the gut. Okay, so for this portion, so this FC portion is required, and then you also require FC portion for activating, for example, your NK cells, the ADCC, and all those things. But it is not required for binding to the antigen. FC portion is not at all required for binding of antibody molecule to the antigen.
Fine, sir. Now got it. Yeah.
But it has other functions, other biological activities. But antigen binding is independently existing of or occurring of FC region. All right. So now there are, I told you about the hinge region, right? But there are two antibodies, basically IgM and IgE, where you actually do not find hinge region. And essentially, whatever the sequence that you have in hinge region, so they actually lead to another constant domain. Okay, so here you have up to CH1, CS2, CS3, right? Here you have CH1, CH2, CH3, CH4 domains. But you don't call it a very flexible molecule because it really does not move around that well. IgM and IgE do not have hinge region in addition to, rather in place of hinge region, so they have an additional domain. Right? That's one. Now, someone was asking that the structure of antibody is shown this way. So, well, I have shown it this way here. So it really doesn't matter. So they can keep on moving around, right? Around these two chains can keep on kind of moving around. Well, but not at the site where disulfide bond is. They will still remain attached, but they can keep on moving around. Okay. So what additional thing that I need to tell you here? Well, every other thing I already have explained. Right. This was the main thing. Yeah. So immunoglobulin fold, and these are called different domains. The name you already know, whether this is present in the variable region or in the constant region, whether this is heavy chain or light chain, you call them as VL or VH, right? You have the carbohydrate binding region, you have the complement activation region, right? We already have talked about all these things. So what is remaining? Yeah, the antibody to the FAV fragment could react with both. Yeah, well, this is an additional thing. So this is actually a part of the structure determination where they did digest it with papain, and then they showed that somehow if you have this FAB and FC, okay, so these are still proteinaceous molecules, these are still protein molecules, and if you can actually generate antibodies against FC portion, anti-FC and anti-FAB, although these are antibody by themselves, but you can actually generate antibody against these protein molecules if they are actually injected in a different species, right? So this is anti-antibody. Okay, now what they are trying to or suggest here is, or show here is that if you generate an antibody against FAB fragment, against this fragment, or against FC portion, so the anti-FAB antibody will show reactivity against both heavy and light chains, because in FAB you have both. You also have basically the light and the heavy. Whereas the antibody to the FC fragment will show reactivity against the heavy chain only, right? And this is basically how it was shown that the FC portion is having only the heavy chain, right? And not the constant, not the light chain. FAB consists of heavy and light chain, and FC contains only heavy chain. Okay, so this is what it means. See, these were the experiments that were done to define if you have four polypeptide chains, basically how they are connected, which portion is showing reactivity to antigen, which portion is not showing reactivity to antigen, which portion is required for binding to the antigen, which portion is required for other biological activity. For example, if you incubate FAB with complement component, so you don't get any binding. If you incubate FC portion with the complement component, you get binding. So all these things are defined by these biochemical analysis, right? Okay, so now this basically suggests the localization or organization of immunoglobulin genes, the genes which encode for the antibody molecule. Okay. Now you could see that antibody is a polypeptide, right? And if it is a polypeptide, so it must be encoded by a gene, and gene is present on a chromosome, right? And I'm pretty sure you must be knowing about one gene, one polypeptide hypothesis, and one gene, more than one polypeptide hypothesis. Yes?
Yes, sir.
But have you also been taught that more than one gene encoding for one polypeptide? Well, there is only one example, and that one example is the antibody molecule or the TCR. Okay, so here you have more than one genes encoding for a single polypeptide. And how is this brought about? So we are going to discuss in some detail. But this is just to emphasize that the immunoglobulin locus for heavy chain is present in humans on chromosome 14 and in mouse on chromosome 12. And for light chain, this is present on a different chromosome. Okay, in human, this is present on chromosome number 2. Here it was 14, 2 and 14. And here, basically, it's 6. Light chain is on 6. And in mouse, this is chromosome 12. They are not even present on the same chromosome to give the antibody molecule the structure it has. Okay, they are actually present on different chromosomes. And obviously, on one chromosome also, you have multiple genes, right? And this is what actually make antibody molecules or the TCR so unique in the whole field of biology, because there is one process that we call as recombination events. Okay, once recombination has happened, only then you make antibodies, and this recombination actually happens at the genetic level, at DNA level. Okay, now for the time being, remember that variable region in human and mouse, there are different genes actually which are shown here. Okay, 40 in human, and in mouse it's 110 different segments. Okay, And each one of those could give rise to the variable region, right? And then you have somewhere in between this diversity region. In humans, so they are 24. In mouse, they are 10. And then you have something that we call as joining region. In human, 6, mouse 4. Okay. And at certain distance from this whole V portion, so this whole thing actually through a complicated process of recombination will then recombine with either this mu chain gene or delta chain gene or gamma, which also come in different types, and then or alpha or D, right? Or basically whatever those 5 isotypes that you have. Okay, so these are the basically gene segments which encode for the heavy chains, right? Mu, delta, gamma, gamma 1, gamma 2, gamma 3, alpha, right? And epsilon is also there somewhere. And for light chain, again, you have the variable, you have the joining V, J, and constant. Constant basically is basically whatever that in light chain. So you have this constant region, CL. Remember? This portion. So this portion is, it is basically, this portion is encoded by so many of those different segments that we have talked about, 14 humans, 110 in mouse, and so on and so forth, right? And this portion basically is encoded by, for example, either kappa or lambda. This portion is encoded by either mu, gamma, delta, epsilon, and whatever else you have, right? All these things here. Okay, and this basically is this kappa region, right? And for lambda region, again, lambda has a different organization than the kappa. So basically, you have all the joining region sequestered or clustered in one group for the kappa, right? But in lambda, if you utilize lambda chain for the light, rather lambda gene fragment for the light chain, so you have basically J and constant, J and constant, J and constant, J and constant. So whether it's J1 constant, J2 constant, J3 constant, J4 constant, and so on and so forth. Okay. This is in humans. Again, as you know, this is on chromosome 6 and 2 and 2 in humans and 6 in mouse. The organization is different. This is just to emphasize that although you have one polypeptide chain of heavy chain and one polypeptide chain of light, but these actually are encoded by different gene fragments. And once there is a recombination process, only and only then you start encoding for either the heavy chain or the light chain. And then heavy and light chain combine together to make you basically the antibody structure. Okay, if now somebody asks you more than one gene and one polypeptide, example is TCR and BCR, right? So the complicated process, how basically this is taking place, so we'll talk about in more detail in coming lectures. Okay, so but for the time being, so I refer to as pepsin digestion, right? Now, papain digestion I talked about, right? Papain has this site which actually is above this disulfide bond. Okay, so you have this disulfide bond connecting these two heavy chains. You have this papain digestion here, which will give rise to FAB, 2 of those, okay, and 1 FC, right? There is another enzyme, pepsin. You find a lot of it in your stomach, okay? So this cleaves below these disulfide bond. And if it does so, you are going to generate a fragment which actually consists of FAB prime and then bracket 2. So because there are two FAB, 1, 2, right? But they are all basically, all this thing is present in one complex. Here, so these are, one FAB is separate, another separate FAB is separate. FC, you will get. Okay. Now, if you actually treat it with pepsin, so because those residues which are recognized by pepsin are also present in the other region of the FC, so you are going to get multiple fragments of heavy chain. Now, if you look at the structure, the whole antibody molecule has basically a size of 150 kilodalton, 150 kilodalton. If you digested it with papain, so you are going to get two fragments, rather three fragments. Two of those will be 45 kilodalton. One will be 50 kilodalton, right? Because these two are going to migrate at the same place in SDS page, right? If you digest it with pepsin, so you are going to get one band of 100 kilodalton and then very small fractions coming from the heavy chain. The other thing that you can do is, because there were a lot of disulfide bonds, interchain, okay, basically within these two chains or within one chain where actually you make this immunoglobulin fold, right? And mercaptoethanol is a reducing agent which is going to basically reduce this disulfide bond, and you separate out these different chains. And if you do so, you are going to get one band, rather eventually you will be a dimeric unit, but because the size is same, so it will migrate closer to 50 to 55 kilodalton for the heavy chain, and for light chain you are going to get 22 kilodalton polypeptide bands, right? So this is how actually the structure of antibody molecule was solved. Yeah, so we will talk about this slide in next next class. Basically you digest and then you resolve it, and then you find out which portion has activity against what, and then this is how the structure of antibody molecule was actually resolved. Okay, so let's stop it here because you might have the next class.
Excuse me, sir.
Yeah.
Sir, the kappa and lambda domain of light chain is only responsible for the variable region and not constant light chain region?
No. They are actually responsible for the constant region of light chain, not the variable. Variable, you have already these different segments.
Okay, sir. And sir, in the this mercaptoethanol reduction and the pepsin digestion that you had shown, sir, in FC region, it was seen that there is increase in terms of molecular weight. There was from 50 to 55 kilodalton. Whereas if we talk about...
Okay, so let's talk about these issues tomorrow. Because anyway, I'm going to actually show that that SDS page with the different segments, and then we'll take these questions at that time, right?
Right, sir.
Yeah.
Sir, quick
Yeah.
question. So what is the syllabus for midsem? Like, will it comprise till?
So whatever we complete by this week. So whatever we complete by this week will be for your first midsem.
So what will be the mode via same classroom, or what will be the mode of, like, the mode of conduction? Like,
well, I don't have any clue as of now. So whatever Dean Acad's tell us, we'll do it.
all right, sir. Yeah, thank you.
Right. I think given the current situation, probably it's going to be an online kind of thing. maybe for 1 hour or something, right? So, but I think you will get to know by the end of this week. Some communication will be made to you. If not by our Dean Academy, then obviously I'm going to tell you what needs to be done.
Sure, sir.
All right.